Okay, good afternoon, everyone. Thank you for joining us for today's Connect Science seminar. As a reminder, the talk will be recorded and you can see this link at the website later on. And also as a reminder, if you have a question, please put into the Q&A function, not in the chat function, but the Q&A, and we will try to answer as many as we can at the end of the talk, and then the speaker will have the remaining questions that can get back to you on that. So it's my pleasure to introduce my colleague, Bill Sellers, as today's speaker. And Bill is currently a professor of medicine at Harvard Medical School and Dana Farber Cancer Institute, and also the director of the cancer program at the Broad Institute in Cambridge. And Bill doesn't really need an introduction. He's had a very impressive career. He started out as a faculty at Dana Farber. We actually started together at that time, and he was one of the pioneers of using genomic technologies to dissect prostate cancer, and also working on a PI3 kinase PTEN pathway importance in prostate cancer. Then he took a detour for 10 years to lead global head of oncology at Novartis, where again, he was very successful with taking some of the drugs to a clinic, or just being, promoting new preclinical studies in that area. Then he came back to Dana Farber at Harvard and has been using the, whatever he learned at Novartis to apply it in the academic setting, also with the goal of trying to improve our cure of cancer and developing better therapeutic strategies. And today, Bill will talk about the discovery of context-specific PARALOG dependence in cancer through combinatorial CRISPR screens. So thank you, Bill, for joining us, and we're looking forward to the talk. I think you're muted.
Yep, just another year of the pandemic and I'll get used to this. Yeah, so first, just by way of disclosure, I have a number of conflicts to disclose, but relevant to this, during my time at Novartis, we worked on an inhibitor of BCR-ABL that's an allosteric inhibitor, and I'll talk about that in the beginning of my talk. So that's the most relevant conflict. I just wanted to note David Livingston's passing. I joined David's lab as my first lab as a postdoc before then moving with Bill Kalin to his lab when Bill got his own lab. And I just wanted to say, what a great time and educational and mentorship experience I had with David. My wife asked me, sort of after the funeral, why I wasn't sadder. And I was thinking back about how great the times were we had in David's lab and how exciting the science was. And yeah, just super grateful to him for all of that. And I hope people who are in their labs experience even 10% of what we experienced working in his lab. I'm sure would be a joy. So one of the things about David at the beginning of lab meeting is you'd start your lab meeting presentation, and about like 3 seconds in, he would say, wait a minute, what's the question? And this always brought us back to thinking about the sort of big picture. And of course, one of the most important questions that we can ask ourselves is, how are we going to achieve cures in cancer? And this is interesting because I think many people don't think we've actually cured cancer, but in some cases we have, but we don't really understand that much about how that's happened. So in general, one thing we could probably say is usually combination therapy is required. So in the 3 cancers that I've listed here, testicular cancer, diffuse large cell B-cell lymphoma, and ALL, empirically derived combination therapies led to significant cure rates in those diseases. And I think people probably forget testicular cancer can be cured when it's widely metastatic, including with brain metastasis. Now, presumably, these drugs all work because they have mechanisms of resistance that are non-overlapping so that the cancer cannot find a single clone that is cross-resistant to the combinations. So the question in the sort of post-genome, let's say precision medicine era, is whether we can start to understand combination effect and the design of combinations based on a genetic understanding of cancer. That's at least the question I'm interested in. So the first thing we have to think about is, where does the resistance occur? And of course, that's because of this regional heterogeneity and subclonal heterogeneity that arrives, that's been so admirably demonstrated by many people. In this case, this is the work from Charlie Swanton's lab looking at renal cell carcinoma and the tree-like evolution, which can be quite daunting. But one of the really exciting aspects on the more positive side was, in every case, in every tumor, the mutation in the von Hippel-Lindau gene was the central truncal event, and it was preserved in every clone. So one can imagine that if you actually had an ability to reverse the effects of the von Hippel-Lindau gene, you would have a significant effect on every clone, versus if you, in this case, targeted PI3 kinase mutations, which are subclonal and heterogeneous. Of course, that would only likely work in those subclones. So then when we think about how to treat cancer for curative intent, one idea is that if we could identify in the trunk multiple required events, we'll call those drivers for now, let's say driver 1 and driver 2, and we could develop inhibitors for those two drivers, and those inhibitors were non-cross resistant, then maybe we would have an approach to curing that cancer. Unfortunately, what's been difficult, it's been difficult enough to find two drivers, and it's certainly been next to impossible to find two drivers where we have two drugs that are sort of very potent against those drugs and used in combination. So what I want to tell you next is this more surprising idea, or I think it was at least surprising, where if you target one driver with two drugs, and those two drugs have a non-cross resistant mechanism, you can make significant inroads against preventing subclonal resistance. And the two drugs I'm just going to briefly describe are imatinib, or really any catalytic inhibitor of BCR-ABL, and ABL001, now called asciminib, which is an allosteric inhibitor of ABL. And asciminib is interesting because, of course, it binds distinctly to imatinib, and this is the actual crystal structure, not a model, showing that the two pockets on the protein can be occupied simultaneously by the two inhibitors. And ABL001 on its own right is extremely potent. It has a 1.2 nanomolar biochemical IC50 and a 1 nanomolar IC50 in a cellular assay. By comparison, Gleevec is actually 200 nanomolar, and sort of the next generation, nilotinib, was 20 nanomolar. So because these two molecules bind simultaneously, we hypothesized that perhaps it would be possible that the mechanisms of resistance would not be overlapping, and therefore the two drugs would be acting independently against the emergence of clonal or subclonal resistance. We did a genetic barcoding to show that they were in fact non-overlapping with respect to the evolution of cancer in vitro. I'm not going to show that data, but we then took BCR-ABL positive KCL22 cells, implanted them in vivo, and if we treated first with a catalytic inhibitor, in this case nilotinib, you can see you got full regression, then resistance, and the resistance mutation that emerged was in the catalytic domain. If you switched to the allosteric inhibitor, you regressed that. So the catalytic inhibitor has activity against the nilotinib resistant alleles, but by virtue of the fact that you had expanded the population again, now resistance develops to the allosteric inhibitor. If you reverse the experiment, you treat with the allosteric inhibitor, you regress the tumor again, and now resistance emerges, in this case, with an allosteric point mutation. If you switch to nilotinib, nilotinib is active against the allosteric site mutations. So again, non-cross resistant, tumors regress, but then they relapse again. If instead you start out at the same point with both drugs, you regress the tumors. We treated, you can see regress to nothing. We treated the tumors for 80 days, then stopped the drugs and let the mice live for another 100 days without evidence of relapse. So in this case, as it pertains to this preclinical model, the combination of these two non-cross resistant drugs attacking a single driver basically cross-eliminate resistant clones and are able to eradicate the tumor in vitro. So as I mentioned, it's been hard to find more than one driver in a cancer, and again, harder to find two drivers where we have drugs. What about the idea that a driver might elicit a new cancer dependency that is not the same as the driver mutation, but is, for example, downstream, and that we could develop drugs against that cancer dependency? So I want to share with you our next approach to this idea, which was taking on the dependency of tumors that have deleted P16 and then co-deleted methylthioadenosine phosphorylase and the resultant dependence on the methyltransferase PRMT5. So we showed in a paper we published from Novartis in 2016 in Project Drive, a project where we targeted around 8,500 genes with 20 shRNAs per gene, that in cells that had deleted the methylthioadenosine phosphorylase gene, which is 100 kb away from the commonly deleted tumor suppressor P16, these cells are selectively inhibited by the shRNA-mediated knockdown of PRMT5. So we were interested in thinking about this from a mechanistic point of view and then a therapeutic point of view. So the work from my own lab in this regard was carried out by Kathleen Mulvaney, with collaboration with Brian McMillan in CDOT, Alessandra Iannari in our lab, a chemist named David McKinney, and a number of research associates in the lab who also participated. So I need to explain to you how this dependency on PRMT5 arises. So again, CDKN2A is the most commonly deleted tumor suppressor. When it's deleted, about 80% of the time, methylthioadenosine phosphorylase is also deleted. This gene processes methylthioadenosine, so if you delete it, methylthioadenosine accumulates in cells and turns out to be a competitive inhibitor with SAM, which is the methyl donor for all methyltransferases. But we went on to show that MTA selectively inhibits the methylation activity of PRMT5 by forming this very unique inhibitory structure shown here, which relies on two residues at the bottom of the PRMT5 pocket that are actually unique to PRMT5. So again, MTAP deletion, you get accumulation of MTA. MTA competes with SAM and acts as a partial catalytic inhibitor selectively of PRMT5. Now, we proposed in this first paper that MTA cooperative inhibitors, that is an inhibitor that would co-occupy the pocket with MTA, would be a really cool way to take advantage of this specific genetic defect. And recently, Mirati Therapeutics published the first, what I think is looking like a bona fide MTA cooperative inhibitor called MRTX9768, and the data look quite compelling for it to exploit this difference. Shown here is the relative IC50s of this inhibitor in the MTAP deleted versus the MTAP wild type cells. So you can see there's a significant selective inhibition of cellular growth in the MTAP deleted setting. So we were not interested in my academic lab in pursuing that. We were thinking that maybe there would be other sites on PRMT5 that would be spots for therapeutic intervention besides the catalytic domain, taking this lesson from the allosteric inhibition of ABL that I just showed you. So what's been apparent in the MTAP deleted synthetic lethality is, in addition to PRMT5 scoring very strongly in the MTAP deleted setting, a number of other molecules also, genes also score in this screen, including WDR77, which is the obligate heterodimer partner of PRMT5, and then two substrate adapters, Rio kinase, which binds PRMT5 and recruits nucleolin for methylation, and PICLN, which binds to PRMT5 and recruits the spliceosome for methylation. So we asked, or we're interested in whether or not the interaction site, if one could identify how the substrate adapter is bound to PRMT5, could this be a site of intervention? And Brian McMillan and Kathleen looked at the substrate adapters, and they identified a 7 amino acid highly conserved sequence in rheokinase, PICLN, and the third substrate adapter, COPR5, that looked highly homologous and was located either in the N or the C terminus of these three substrate adapters, and then asked whether or not this peptide was sufficient in SPR biochemical assays for binding to PRMT5. And as you can see here, the three different substrate adapter sequences embedded here all had robust binding by surface plasma resonance. Now, Brian went on to solve the crystal structure of the substrate adapter peptide bound to the heterooctamer of PRMT5, which is shown here. So PRMT5, shown in pink, actually is not a single molecule. It's composed of 4 PRMT5 subunits bound to 4 WDR77 subunits. And then you can see in this structure, 4 different substrate adapter peptides are bound to PRMT5. If we look just at the monomer, so here's one of the monomer subunits, you can see the SAM binding site where MTA would be binding in the MTAP deleted setting is shown here. The substrate adapter molecule is distant from this pocket. So these two sites are distinct. And again, if we think about the fact that MTA might be occupying this pocket, we're interested in the idea as to whether or not disrupting this interaction might have therapeutic consequence. So this is the interaction site at a higher resolution in the crystal structure. And based on this, we asked now 3 questions from the biology point of view. Is this binding site on PRMT5 required? Is it intact binding site required for the methylation of PRMT5 substrates? Is this site, which we call the PBM groove, a druggable site? And the last and most important is the PBM groove required for the viability of cells in the MTAP deleted or deficient setting? So to do that, Kathleen introduced 3 key mutations and 3 key residues. These are residues on PRMT5, not on the peptide. She then knocked out PRMT5 by CRISPR and simultaneously reintroduced this so-called ADA mutant, and along with either reintroducing the wild type PRMT5 or the catalytic dead. And you can see the catalytic dead reintroduction wipes out methylation and doesn't restore it. The wild type after the knockout completely restores the methylation of PRMT5 substrates, as shown in this SDMA Western blot. Interestingly, the ADA mutant actually has a partial phenotype. There are some substrates, like this one shown at 25 or 15 kilodaltons, that seem to be ADA dependent. That is, they are dependent on this mechanism. And then there are other substrates which seem relatively independent of this. So based on this data, we now think there are substrates of PRMT5 that are being recruited through the substrate adapter mechanism, and then likely substrates of PRMT5 that are not. This is shown a little more clearly here, where we look specifically at certain substrates of PRMT5. So I said nucleolin was a substrate of rheokinase 1. You lose that methylation when you delete the binding or mutate the binding site on PRMT5. And similarly, a substrate for COPR5, namely histone 4, its methylation is lost when you mutate the site on PRMT5. But as you can see for this substrate ZNF326, actually its substrate methylation goes up when we make a mutation in the PBM groove. So there are clearly distinct substrate recruitment mechanisms by PRMT5, several of which go through the canonical pathway of substrate adapters, and one at least exemplified by ZNF326, which is independent of this site. So to get to the second question, is the PBM groove a druggable site? Here we were lucky enough to collaborate with the Center for the Development of Therapeutics at the Broad and Deerfield Management to develop an HTS assay based on a displacement of this peptide that I just showed you. That would be a fluorescence polarization displacement assay. We also, with the structural information, conducted a virtual screen. And then again, based on the structure, we also conducted an NMR screen. In total, we screened roughly 800 to 900,000 compounds, and in the end had exactly one validated hit that passed our flowchart criteria. And that's shown here. This was a relatively weak hit is as common in initial screens. And you can see here it displaces or inhibits binding of the PBM peptide in this fluorescence polarization assay, but not an irrelevant peptide interaction consisting of MCL1 and NOXA shown here. We went on to solve the crystal structure of this small molecule, and in fact, it does bind exactly in the same site as the peptide. But what's interesting is it uses a very unusual warhead to create a covalent link to a nearby cysteine, and thus this is a sort of novel covalent inhibitor of the substrate interaction domain, which we demonstrated by showing a modification of the protein equal to the molecular weight using intact mass spec, which did not occur if the cysteine was mutated to alanine, as shown here. Now, we've done some limited chemical optimization. This is quite a challenging scaffold to work on. In addition, I would say from my experience at Novartis, having only one scaffold to work on from a drug discovery project perspective is not ideal. And we were able to, this compound not being the most cell permeable compound, we were able to show some evidence shown here and here of disruption of methylation and disruption of the interaction between the substrate adapters and PRMT5 using a nanobit assay. Now, is this site required for the viability of cancer cells if you delete PRMT5 and restore this mutant function? These experiments were technically challenging to do. And in the end, we were able to show while restoration of the catalytic dead PRMT5 significantly impaired the growth of reconstituted cells, the so-called ADA mutant had a viability effect, but it was nowhere near as significant as the catalytic dead. So we're not 100% sure why this is. It could be that there are compensatory substrates that are making up for the loss of the canonical adapters, or it could be that trace levels of the endogenous PRMT5 that we can't eliminate through selection are also contributing to the viability effect being more modest than we would have anticipated. We also went on to do colony formation and xenograft assays, and you can see there is an effect in vivo, but again, it's a retardation of cancer growth, not a regression of cancer growth. So to summarize this part of the talk, the PBM groove on PRMT5 is required for the recruitment of adapter-mediated substrates, but not all substrates go through this mechanism. And Kathleen's quite interested in pursuing how these alternative substrates are recruited, for example, CNF326. The PBM groove is potentially druggable through covalent inhibition. However, to date, the mutation of the PBM groove, and we only have sort of partially optimized tool compounds. Those tool compounds, either mutation of the groove or tool compounds only result in a partial loss of viability in cancer cells. So we are now going back to see whether or not these mutants can cooperate with the Mirati MTA cooperative inhibitor to see if we can unveil a greater viability effect in combination with those inhibitors. So this is wrapping up the PRMT5 side, and I would say it's fair to say while we still have this idea that there are two sites on PRMT5 to target, the MTA cooperative site in the catalytic domain being one which is most advanced, and this being still a tentative and hypothetical site for further exploration. So going back to this idea of how do we cure cancer, I told you about this ABL001 story of two drugs targeting a single driver. But another possibility is maybe in addition to having a driver, maybe a driver would actually create two pathways, both of which were required for maintenance of cell viability in a cancer cell. And therefore, if you had drugs to both pathways, then you'd be able to, again, have a rationally designed, non-cross-resistant therapeutic combination. So finding these dependent states has been the objective of large-scale screening efforts, either the shRNA or CRISPR screens, the CRISPR screens being carried out at the Broad and the DepMap project and at the Sanger Center and other places. And what's been clear is some very interesting and exciting targets have emerged from that. So for example, I mentioned PRMT5, but also the discovery that the Werner helicase is synthetic lethal in mismatch repair deficient colorectal, endometrial, and gastric cancer is also quite exciting. But what's kind of interesting is we haven't found a ton in the major oncogenic pathways. So for example, if you look at PTEN wild type versus none, we don't really see new dependencies. If you look at, BRAF wild type versus mutant, we don't see a ton of new dependencies. And so one has to start wondering, is that it? You just have the oncogene and there's no dependency downstream, or are we missing something? And so obviously I'm interested in the idea that we might be missing something. And one thing we're clearly not capturing is the idea that in these important oncogenic pathways, there is significant genetic redundancy. So I like to draw the RAS pathway like this because it's much easier for me to understand. But in reality, if you want to look at paralogous gene function in that pathway, this is more like what the pathway looks like. Still linear in this case, but with built-in redundancy at nearly every level. And that's important to take into consideration because we know that you can make inhibitors that inhibit closely related paralogs. In fact, I'm not sure it's possible to make a MEK inhibitor that selects between MEK1 and MEK2 or an ERK inhibitor that selects between ERK1 and ERK2. Instead, we have MEK inhibitors and ERK inhibitors. But when we go to do the CRISPR screens, we can knock out KRAS and show like a whole bunch of cancer cells that are mutant for KRAS care about KRAS. But when we try to look at MEK1 or MEK2 or ERK1 or ERK2, it's very hard to see the dependent state downstream of RAS by just looking at the single gene knockouts. And so the question is, what would have happened had you targeted both MEK1 and MEK2 or ERK1 and ERK2 or pairs of RAF genes downstream of RAS? So that's the idea of trying to find context-specific paralog dependencies. And this project was led by Takahiro Ito in the lab, along with a fantastic computational scientist, Madi, and a number of research associates, Michael and Diego, and then Rui Tong and Sid, who are also computationalists working on this project in collaboration with our colleagues in the genetic perturbation platform at the Broad. So again, the question is, do functional redundancies or mask otherwise druggable vulnerabilities? And I already mentioned this idea that inhibitors can inhibit two closely related paralogs, but CRISPR screens are only targeting one of the two pairs. So we're interested in creating a combinatorial CRISPR screen that would target both closely related paralogs. So how to do this, it's not as simple as just taking two guides and sticking them into a cell. And part of the reason for that is if you use guides that have the same tracer sequences and or the same promoters, they undergo recombination either in your synthesis of the vector or during lentiviral infection or later during PCR amplification for sequencing. So to avoid that, John Dench in the genetics perturbation platform created this system called Big Poppy, in which he engineered two different Cas9 tracer sequences, one for Staph aureus Cas9 and one for Staph pyogenes Cas9, driven by the U6 or H1 promoter. And the advantage of this is that these two tracers are different and therefore there won't be recombination. So we chose to use this system for the PARALOG library. And the design of our library was to use 6 guides for each paralog. So 6 guides for paralog A, 3 driven off the pyogenes side, 3 driven off the aureus side, and then 6 guides for paralog B, again, 3 from the pyogenes side, 3 from the aureus side. And then these 6 guides, the blocks of 3, would be combined into 2 blocks of 9, giving us 18 guides in the combination. So when we look at data for pairs, we have both the single gene knockouts targeted with the paralog A guides in this case, along with AEVS as a cutting control, and then vice versa. So here's what the data looks like at a granular level for BCL2L1, BCL2L2, and the pair. You can see this is an example of synergy where the 6 guides to BCL2L1 have no effect, the 6 guides to BCL2L2 have no effect, and only the guides in combination targeting both genes have a viability effect, showing that there's functional redundancy between these two paralogs. I won't go through the cloning of this. There's some tricks as to how we created the library in the first place. This will all be coming out in Nature Genetics, I think, hopefully shortly. Now you can tell we have 30 guide vectors per paralog pair. That means we're not going to be doing whole genome paralog screen. So we have to sub-select the families of paralogs that we want to target. And in this case, of course, we focused on enzymatic classes like kinases and phosphatases and methylases and demethylases as our first foray into this. And we created a library that targets roughly 5,538 unique paralog pairs and is screened in kind of the standard way. First, the cells are engineered with the Staph pyogenes Cas9. Then the paralog library, which on the vector is included, the Staph aureus Cas9 is introduced into cells. We grow those cells for 21 days, harvest genomic DNA, PCR amplified. And then in this case, we want the sequence of the 2 guides together. So this requires paired end sequencing. So to evaluate the performance of this library, we screened an initial set of 11 cell lines and had certain genes included as positive and negative controls. So we have non-essential genes paired with the AEVS cutting control. You can see their distribution is shifted to the no effect side. We had included pan-essential genes, for example, CDK1, along with an AEVS cutting control. And you can see the single guides are significantly shifted to the negative log fold chain, suggesting that single gene knockouts are working. Now, we don't fully know which paralogs are not lethal versus which ones are, but we guessed that certain paralogs that have no or very low expression would be more likely to be non-essential, and those are in fact distributed more evenly across the cell lines in terms of log fold change. And there are some paralogs which we know from single gene knockouts when one paralog is deleted and you do a CRISPR screen and the second paralog is deleted, that it's lethal, and that set of genes is likely to be a pan-lethal set. For example, BRG and BRM are a pair like this. And you can see these are significantly depleted compared to the non-express controls. So this is preliminary evidence that the library is working. As an example, you can see what the screen looks like for a paralog pair that we think is lethal, which is HDAC1 and 2. So HDAC1 by itself, not very effective. HDAC2 by itself, not very effective. And then HDAC1 plus 2 kills almost all cells except for MEL202, which is in fact impaired by HDAC2 by itself. And that's because MEL202 doesn't express HDAC1. So these are sort of some evidence that the library is performing as expected. This is an example of what the data look like, plotting on the 11 cell lines on a Manhattan plot to give you an idea of how many new lethalities you see above the single gene knockout. So in every case, the red dots here are genes that are scoring with we have an algorithm we developed called Gemini. The Gemini score is a measure of synergy. This is the false discovery rate for the combination effect. So there are quite a few hits being missed in the single gene knockout screens as sort of shown here. I wanted to illustrate one kind of cool example from a drug discovery perspective. In the cell line set, we included one uveal melanoma cell line, and we included that because uveal melanoma is driven by mutations in GNAQ and GNA11, and these are upstream of PKC, and there's a number of studies that have said PKC and PKC inhibitors are important therapeutic notions in uveal melanoma. So when we look at this one uveal melanoma cell line, you can see what our PARALOG library looks like for PKCs. Literally, like every combination of PKC with one isoform with the other is included in the library. So in reality, we're screening mini libraries against families of paralogs. And what's interesting in uveal melanoma, if we blow this up, is uveal melanoma has a strong dependence on PKC compared to the other cell lines. But in every case, PKC epsilon is a component of the pair. So while these are synergistic, suggesting that there's some redundancy with multiple different PKC family members, we can see that PKC epsilon is the dominant isoform, meaning you really want an inhibitor that's going to be, in this case, dominant on epsilon, but it may be the case that you also want activity against other PKC isoforms to prevent escape if these data reproduce in other uveal melanoma lines. Now, we didn't have a ton of cell lines, and that means we don't have a ton of power to look at context-specific lethalities. But as a first foray into this, we included 4 NRAS mutant melanoma cell lines and 2 what I would call wild-type melanoma cell lines. One is truly wild-type, one has an NF1 mutation, which in many cases could activate the NRAS pathway. But nonetheless, we took the 4 NRAS mutant cell lines and compared them in a volcano plot to the 2 non-NRAS mutant cell lines and looked for pairs of genes that had a significant log-fold effect and synergy in the NRAS mutant setting, since really no one has found synthetic lethal nodes in the NRAS setting. So one interesting thing here is that you can see BRAF and CRAF are scoring, which is interesting because CRAF in the mouse is critical downstream of KRAS, as shown by Tuvicin and Barbacid. Here you can see there's not a lot of synergy by the size and color of the dot, and that's because actually CRAF is a dominant isoform, dominant enough that the synergy score is relatively modest. From a synergy perspective, 2 14-3-3 proteins scored, epsilon and zeta. It turns out these 2 14-3-3 proteins are the ones that assemble the BRAF complexes and are required for BRAF activation. And these look like a novel paralog pair in the NRAS mutant setting. And then finally, CDK4/6 and DUS4/6 are also hits. And we focused a lot of our attention on DUS4/6, which you can see here has very little effect in the MAP kinase wild type or NF1 mutant cell lines, but shows significant synergy in the NRAS mutant setting. So we went on to validate this in single knockout studies, meaning creating the double knockouts, but doing them in cell lines not in a pooled format. And here you can see 2 NRAS mutant cells where neither DUS4 nor DUS6 have any activity on their own, but the double deletion impairs cell proliferation in single experiments. Now, what's interesting is, of course, why not BRAF mutant cells too, right? And there's no reason that it couldn't be. We just hadn't screened any. So we went back to the Ivana single gene knockout data, looked at BRAF mutant cells, and what you can see is there's a small subset of BRAF mutant cells that actually have a single gene effect with DUS4, while the majority of BRAF mutant cells have no effect with DUS4 knockdown. If we look at DUS6, none of the BRAF mutant cells, or for that matter, the NRAS mutant cells are affected by DUS6 depletion. And just going back to the NRAS mutant cells, none of them are singly affected by the DUS4 deletion. But this raised the question as to whether or not DUS4/6 would be more effective than DUS4 in the BRAF mutant setting. So TOC went ahead and picked 2 cell lines, IGR1, which is right in the center of the unaffected set in the DUS4 data, and WM2664, which is sort of at the -1 log-fold change. And again, repeated these experiments with the single and double knockouts. And you can see that WM2664 with DUS4 has some viability effect. DUS6 has no effect, but the double deletion again has a more profound effect than the either single deletion alone. And then in the IGR1 cells, neither DUS4 nor DUS6 guides have any effect, but the double knockout has a significant effect. So this looks like evidence to suggest that DUS4/6 is a paralog dependence in both the NRAS and BRAF mutant melanoma settings. And that's interesting because that's about 70% of all melanoma. So how does this work? Now, many people might know that DUS4 and 6 are not activators of the pathway, they're inhibitors of the pathway. That means if we're knocking them out, the pathway should be actually turning on. That's somewhat counterintuitive. So let's see what actually happens. So we knock out DUS4, you can see the levels of DUS6 actually go up. So there's some compensation that happens acutely. If we knock out DUS6, DUS4 goes up, so there's compensation in that axis too. But if we knock out both, of course, there's no compensation by either one of them, and instead we get significant elevation in phospho-ERK, suggesting that instead of inhibition of the pathway being lethal or toxic to the cells, that in this case, it's ERK hyperactivation that's impairing viability. So to test that, we asked whether or not an ERK inhibitor could rescue viability in the setting of DUS4 or 6 depletion. And so these are the same experiments I showed you before where we're depleting DUS4/6 by double knockout, but now we add in the ERK inhibitor shown in beige here and in the NRAS mutant setting and in the BRAF mutant setting. Double knockout of DUS4/6, the effect on proliferation and viability is rescued by an ERK inhibitor, suggesting that the mechanism by which this happens is through ERK hyperactivation. So to summarize so far, DUS4/6 is a paralog dependence. It's likely relevant in both the NRAS and BRAF mutant setting, and dependence on DUS4/6 results from the loss of negative regulation and hyperactivation of ERK. And this goes back to models that others have talked about. In fact, we showed some evidence of this in a 2012 Nature paper where we showed BRAF inhibitor withdrawal in vivo causes hyperactivation and tumor regression. This is, again, consistent with this idea that there's an optimal zone for fitness. Some people call it the Goldilocks zone of ERK activation, and either inhibition through BRAF MEK inhibition or activation through DUS4/6 inhibition impairs viability. So the next question we wanted to ask is, well, what happens during resistance to inhibitors? Because theoretically, when inhibitors are treating cells, cells need to forcefully activate ERK to overcome the inhibitors, and that might make them more sensitive to further activation. So to test that idea, Corey Johansson at the Broad had been doing experiments where he was generating BRAF mutant resistant cells resistant to either dabrafenib, trametinib, or dabrafenib, trametinib, and the Schering-Plough ERK inhibitor. And then after creating these resistant screens, doing a CRISPR screen, in this case, just single gene knockout studies. So the first thing you can see is that when you make these cells resistant, the basal level of ERK definitely goes up, as you can see here. And what's interesting also is DUS4 is also going up, presumably keeping this in control. And now when you do, the genomic screen, the very top most increased dependence between the parental cells and the resistant cells is DUS4. So as cells become resistant, they become increasingly dependent on DUS4, single gene knockout in this case. So Tak went on to look at, again, DUS4, DUS6, and the DUS4/6 double deletion. And you can see in the parental cells, as was predicted by Corey's screen, there's no dependence on DUS4, but DUS4 becomes a dependence as cells become resistant. The parental cells, as we suggested, are in fact dependent on DUS4/6, as evidenced by the impaired viability of the double deletion. But as they become resistant, they become more susceptible to depletion or more affected by the DUS4/6 depletion. So this, to summarize this just last part, resistance to current BRAF MEK inhibitor treatment increases sensitivity to DUS4 depletion. And vis-a-vis the model I just showed you, you can imagine you're still within the optimal zone, but now the basal activity of ERK is higher, so that when we inhibit DUS4/6, the level of ERK activation is even higher, and they're more susceptible to that impaired viability consequence. So what's attractive to us about this is that we think we have one driver, let's say NRAS or BRAF, and it's actually causing two different types of dependence or vulnerability. It's vulnerable to ERK or MAP kinase pathway inhibition, as evidenced by the therapeutic efficacy of BRAF and MEK inhibitors, but it's also susceptible to activation of ERK. And what's I just showed you is that as you become resistant to an ERK inhibitor or a MAP kinase inhibitor, you become more sensitive to an activator. So this suggests not only would a theoretical regimen of inhibition followed by activation not be cross-resistant, it would actually be cross-sensitizing. So we're quite interested in the idea that ERK inhibition, MAP kinase inhibition, would bottleneck a cancer towards a state of more uniform susceptibility to ERK activation through this cross-sensitizing mechanism. And that's the type of work we're trying to do in the lab now to see whether or not, for example, using genetic barcoding, we can show both non-cross resistance and elimination of distinct barcoded populations in sequential, not concurrent, treatment with ERK inhibitors and in this case with synthetic activation of ERK. So in summary, I think we can start to see or imagine that we can see rules for thinking about how to assemble combination therapies. I've shown unanticipated examples of combinations involving two molecules targeting the same protein that are non-cross resistant and one target, in this case BRAF or NRAS, eliciting two different vulnerabilities: a requirement for persistent ERK activation and a sensitivity to ERK activation. And if these results were right, it would result in a cross-sensitizing therapeutic strategy. Finally, the other big point not to miss is that paralogous genes are encoding druggable redundant targets, and these are missed by single gene knockout screens. So we're quite excited about the future ability to discover additional druggable paralog dependent nodes. So I'd like to thank you for your attention. I'm happy to take questions. Our lab is definitely recruiting, and yeah, you can follow us on Twitter, though I have to say I barely know how to use it, so it might not be the most exciting Twitter feed. And our lab website is there, and if people are interested in postdocs, definitely send me an email.
Okay, thanks, Bill, for this very nice presentation. We have a number of questions, and again, as a reminder, please put your question to the Q&A, not into the chat. So the first question is for the first part of the talk in PRMT5 is does drugging the PBM site also inhibit PRMT5 in a non-MTAP deleted cell lines?
So it will impair methylation in those cell lines, but the viability, like as you could see by the effects, the viability effects are quite modest to begin with. So there's really no viability effects that we're able to see in the MTAP wild type setting. But again, the real problem is there's just a modest or hypomorphic effect in the deleted setting.
Mm-hmm. Okay, and there are two questions that somewhat relate to my question also, is that how do the screens relate to the in vivo dependency, considering that many of these pathways are so much influenced by the microenvironment and, what these interactions, are they validating in vivo?
Yeah, so actually DUS4/6 did validate in vivo, but what's interesting is we didn't get that data until the paper was accepted. So it's not included in the paper, which is a little weird, but it did validate in vivo. And maybe the general question is like, how much of in vitro dependence does validate in vivo? And clearly it's important to go on and do the in vivo validation experiments. And yeah, there are definitely metabolic and media alterations in vitro that alter and create false positives. So, there's a lot of sterol and fatty acid things that are, basically seen in vitro because of the fetal calf serum and other things that are artificial in those settings. I'd say the cell autonomous ones turn out to be relatively reproducible in vitro for the most part. I would say that the challenge in vivo is, if you think about knocking out something that is required for viability, by the time you generate the knockout cells and get them into the in vivo setting, you're already starting to select against the phenotype, the very phenotype you're looking for. And of course, if you try to make inducible shRNAs, that's better, but they're leaky too. So, and then when we were trying to do this with 2 knockouts, we basically had to generate CRISPR knockout of DUS6 and 4, and then shRNA inducible knockouts of the remaining PARALOG. So that, there's a lot of technical challenges to doing that experiment well, so that we understand, like, is this really impairing viability or just modestly impairing viability? So I think that's where we are starting to explore chemical induced degrons as the primary means to do in vivo experiments.
Yeah, and then you're not even considering then not even cell autonomous, but the neighboring cell, because in Drosophila, for example, for RES, there was some very interesting findings that RES, was somehow the neighboring cells had an effect on the RAS mutant cells. So anyway, so that would be a different level of trying to develop screens where you have the cellular interactions, dependencies, but anyway. All right, so another question related to the 2 questions actually about what are the targets of DUS4 and 6 that could be explaining the mechanism of death?
Yeah, so the most likely one is ERK itself, right? That these phosphatases dephosphorylate ERK and keep ERK in a relatively inactive state. And in the paper that will come out, we showed that the interaction site of DUS4 and 6 with ERK, which has only been partially solved, so a peptide sequence called the Kim peptide from DUS4 and 6 has been crystallized bound to ERK. It turns out there are naturally occurring activating mutations in that site. And we showed that those also activate ERK. And when you put those into cells, they create a sensitivity and a viability effect. So we think that though we haven't proved this, the likely scenario is DUS4/6 depletion is depleting the phosphatase activity against ERK. And by the way, we did show the phosphatase activity was required by reintroducing the C to S phosphatase dead mutant, but that's the most likely substrate.
Okay, so related question to that is like, what is the status of DUSP inhibitors? Like, are there any DUSP inhibitors in development?
So there are something I consider not a DUSP inhibitor that's called a DUSP inhibitor, and that's been published. So there is something published as a DUSP inhibitor, but I don't believe that's really a bona fide inhibitor. So that's the extent to which I'm aware of DUSP inhibitors. People might know the work we did at Novartis on SHP2 inhibitors, where we discovered allosteric inhibitors of SHP2. If I were going to go directly after DUS4/6, that would be the approach, because the phosphatase catalytic domain is an extraordinarily highly charged cavity, which has been like notoriously hard to drug. But as I mentioned in the paper, we also showed this interaction site between ERK and DUS. And we are sort of thinking about whether that's a therapeutic approach targeting the surface of ERK rather than DUS directly.
Okay. And another question by Megan is about, do you suggest that the pan-RAF inhibition is not a good strategy for NRAS mutant melanoma?
No, I think the BC RAF inhibitors, though I'm not 100% sure about whether ARAF will ultimately compensate for the loss of B and C RAF. I think BC RAF inhibitors are very interesting. And yeah, there's another conflict, which is we made one called LXH254 at Novartis. And so it'll be interesting to see clinically how those pan out.
Okay, and 2 questions related to the CRISPR screens. So Kai, from our Dana Farber colleague, is like, how do you sort out which pairs of paralogs represent promising therapeutic targets versus rather than liabilities for greater toxicities? Because, you know, you hit out 2 things and they could be killing.
Well, so if the question is designed at like, once we've made the library, how do we evaluate things that look toxic versus things that are selective? That's relatively straightforward, which is the things that are pan-lethal, like HDAC1 and 2 are pan-lethal, you know, are relatively less interesting to us than the ones that show selective lethality. But the reality of that is we need ultimately larger cell lines, a larger number of cell line screens to more robustly distinguish pan-lethal paralog pairs from the selectively lethal. So that's like the first level. I would say a secondary level that we have both with single genes and knockouts is there are things that are probably pan-lethal. It turns out CDK4/6 depletion is pretty anti-proliferative as a paralog pair, except in the RB mutant cell line, right? So most cells are going to be susceptible to CDK4/6, yet we can make CDK4/6 inhibitors that work in vivo. So that means CRISPR experiments are going to miss things that, when partially inhibited or temporally inhibited, could be selective therapeutic targets, which if just CRISPR knocked out would look pan-lethal. So that's like, you know, that's something that really is not being addressed. It was kind of addressed by the shRNA screens, but I think this will be important to, you know, figure out downstream.
Yeah, another related to the CRISPR screen feasibility is like, what is the feasibility to do not just the paralog double knockout screens, but origin by origin permutation double knockout screens?
Well, yeah, that's totally impossible. So at the moment, yeah. I mean, I think I didn't get to show, I just want to see if I have no, sorry, yeah. I didn't get to show, you know, not everything is perfect in these screens, right? And maybe I can show here. So if we look at this cluster right here, you can see this beginning of a bifurcation of the 2 groups. And what's happening is every time there's a guide, let's say this guide over here doesn't work, it gets paired with 3 other guides. And if there's a guide over here that doesn't work, it gets paired with 3 other guides. So people think, oh, we know a guide, we're just going to make 2 guides and that's going to be it. No, like you would need to know a guide actually works. So since you don't, if you want to have negative results that are meaningfully interpreted, meaning you know you've knocked down the pair, you need to oversaturate the screens compared to single gene knockouts. Hence, we're using 18 guides per pair. So if you imagine what that would be like at the 20,000 by 20,000 genome scale library, yeah, that's 20,000 times 20,000 times 30. So yeah, I'm not anxious to do that screen anytime soon. So.
Yeah, you would need a factory for tissue culture then to do that. So.
From gigantic bioreactor, I think.
Yeah, we have 2 more questions. Let's try to do that. So first one is, how did you determine a combination of analog genes in a CRISPR screen? For example, there are many other DOS proteins. Why did you pick 4/6 instead of 10?
Okay, so in the DOS family, yeah, I didn't show here. I'll go back to the. Let me go back.
I mean, basically, how did you pick the paralogs? Because sometimes there's more than 2 for some.
Yeah, the paralog. So, but this is a pretty good example. I don't know if it shows up, but this is like the PKC family. So you can see once we're in a family, we're targeting almost every possible pair. In reality, we used a cutoff of about 30% homology. And in the DUS family, what happened by using that cutout is all the DUS pairs were covered except 2. And so yeah, that what turns out we are now we have a version 2 library we've made. And so now once we pick a family, we eliminate the cutoff and we do an all by all within the family. So, you know, within a subfamily of the kinome where you have a lot of paralogs, we wouldn't bother to create the cutoff. We would just do like every pairwise comparison. So in the new library, that's what it was. In the first library, it was around 30% homology.
Mm-hmm. Okay, and the last question is, did you see enrichment in a Hippo pathway in your screen? Because Hippo pathway has been shown up as a, mediator of resistance to many different agents.
Well, we didn't do resistance in the PARALOG screen, and whether it came up in Corey Johannessen's screen, I don't know. I would have to look, and presumably that data will be available to look at.
Okay. All right, lots of questions from the audience, so at least they paid attention. Okay, thank you so much. Thanks, Bill, for a great talk, and thank you everyone for joining us today. Thanks. Bye-bye.
Thanks for having me.